then I've got to, where's my, here, we're fine here, so I'm getting back. Okay, so let me bring Cytoscape back. I'll close all the goodies I've got here. So let's close. Okay, okay, and let's open recent. Okay, so this is day two of our advanced cytoscape workshop. We'll pick up where we left last time, which is the network we created with some expression data overlaid on the network of interactions in East. And just to recapitulate, uh, this network you see here was created by uploading the files that were provided yesterday. They were, and they still are, in a day one. Uh, well, this is the wrong place. So let's kill this one. They are provided in a Google Drive under day one. So what we essentially did is we took a file describing the connections in between proteins. So those are two columns. First one and third one are identifiers of the proteins or genes that are interacting. So those are pairwise interactions between two molecules essentially. In the center column, there's a interaction type, and we had either PP corresponding to interaction between protein on the left and protein on the right, or we had PD, which is interaction between protein on the left and DNA, meaning the gene coding for this particular protein on the right. So this was one file that we used, and then the other file was a file that had here our expression data. The first column was identifier of the protein or of the gene, and then there were several columns with information about what was the change in expression for a given protein or given gene in a certain conditions. They always came in pairs. There was a column with the log ratio of the expression in a given condition versus or relative to some reference state, and then there was a number telling us what the significance of the change, how reliable the result is. So this was the other file. Well, there was a third one. We wanted to create the human readable stickers for the genes, so we had another file here that was essentially listing once more identifier of our protein, and then a column. There was a column with the human readable, more informative gene names on the right side. So those three files were used to create the network we've got here, and then I went through the trouble of showing the expression data as colors and thickness of the frame around the labels for given names. I also use the human readable, the meaningful names of the genes to show up as the labels for the stickers. To get, in case you want to check how this thing is done, you can walk through the recording from yesterday, and there's step-by-step procedure which gets all those elements shown here. So this is how we get to the network you see here. Now, the files were given from outside. The files were mysteriously provided in directory. In real life, it's more complicated. The reason is not this one. The reason is in most of the cases you will have access to the wrong one. You will have access to file like this. This is gonna come from your experiment, or someone interested in data analysis will provide you a file that looks like this, more or less, with the rows corresponding to particular proteins or genes. There will be some sort of identifier for the protein or for the gene, and then a bunch of numbers telling reporting the results of experiments. Let's say change in expression in case of expression data, methylation level in case of NGS sequencing, or whatever might come out of experiments. So files like this that list experimental data for each protein will come with experiment, and this normally is available. The problem is the moment you want to map the results like here on the network, you've got to create this network somehow. In the case here, we did have a file, this one here, which actually described the topology connectivity between different proteins. This was provided for the sake of the example. Okay, the in real life, the networks got to be somehow created from scratch. And I did show yesterday one way of building it using the psychic interface here. So now let's let's try it once more. So psychic uh, is a service provided by databases that curate protein-protein interactions, that allows one to actually query all those databases in a similar way, in a consistent way for the information they've got. The way it works is we provide a query to the psychic. Okay, psychic is, by the way, a clever abbreviation of the full name of the service that some programmer at DBI came up with like several years ago, and it's got stuck. Okay, so that's the funny name, but otherwise it provides you a way to query databases for information about interactions. Here's Let's make it simpler. Here, let's just list three, or maybe let's make it gal 1, gal 2. Let's provide 5 names of the genes that we're interested in. Let's say, and then let's try to query the database for the psychic for those. So when we start to query, we're gonna get the window popping up, and now we're gonna have a window showing up with that lists the different databases with information related to those genes. Okay, and at the top you're gonna have one or more sources where information is provided, is available, and then there's gonna be more of the databases that might not have any information related to whatever we're asking for. Now, as you can notice here, we've got a lot records found. The reason is some of those names are pretty generic, and there's more than one organism that has data about gal4. It might be gal4 in yeast, but might be gal4 in several other organisms, and all of these things contributes to half a million of entries for just from intact database alone. So to limit the search to specific organism, and we're interested in Saccharomyces in yeast, we can make the query a little bit more complicated. We can put the list of genes in brackets, and we can ask now for information about those genes or those proteins, and we make sure that they come for yeast. Okay, so this way our query, when you now run it, will have way less records, because now the records are about one of those five genes, but only if they come from yeast. So that's the way to limit the search in Scikit by species. And just to explain one or clarify one more thing, when we list a bunch of keywords, one bunch of strings, here there's an implicit or here. So we're actually asking for something like this. We're asking for any record in Scikit that mentions gal 80 or gal 4 or gal 2 or gal, let's make it something else. Well, we can make it gal, let's say make it gal 11, and also mentions these. This is a way to limit the search to yeast. You can in principle use any species name here. So you can search for yeast, you can use search for human, you can use for, you can look for E. coli, you can look for whatever organism you're interested in at the given moment, and this is way to restrict the queries to subset of records. I'll in a slides for today, I'll provide pointers to where to find details, more details about querying Scikit and what are the ways of querying it. So there's gonna be more, but we're using here just the simplest possible thing. Just we're looking for few genes or few proteins that came from yeast. Okay, so let's search once more. So now, uh, now I messed up something. Let's see why. Why is that? That's strange. Let's search. That's curiosity. It shouldn't behave this way. Let's try once more. Or gal 11 and these. Okay, there was a typo somewhere here. So here we've got 1, 2, 3, 4, 5 genes that are supposedly coming from yeast, and we've got now 1, 2, 3, 4, 5 different databases reporting they've got records related to those. So let's let's pick up IMAX. I prefer IMAX. This is a collective resource where several databases curating interactions contribute to. So it's more convenient to query IMAX other than looking for intact, mint, and whoever else might be contributing. Essentially, what's coming in intact and mint is included in IMAX. So we're gonna stay with IMAX. In the slides I provided yesterday, the intro slides, there's a short section at the end mentioning IMAX and providing few pointers to IMAX. So there's more information there, but on operational scale, we can just pick up IMAX as the source and then import the records. It's gonna take a moment to get those things. We are at this moment querying the databases, so there's gonna be certain delay. It takes time to prepare the data set and generate it. Okay, and then I don't want to merge. I just want to create a network from whatever came from the IMAX. Okay, so I'm gonna just close here, and now I've got the result of my IMAX query shown in a in a standard way. Okay, so let's look at what we've got here. Well, we've got a porcupine here, and we've got another porcupine, and we've got a lot of loops here. Let's rotate it a little bit. This is the way to manipulate the networks. We can pop up the layout tools, and then we can rotate the whole thing. Okay, and now I can zoom. Let me get chat up so I can see if you're asking any questions. Just a moment. Where's the chat? Okay, here we are. Okay, so I'm back here. So here's the network we got in. Okay, as I mentioned yesterday, each record that came from IMAX corresponds to one particular experiment that demonstrated a given interaction. So if we look at the two proteins here that ended up connected by three lines, three edges, it means that there are three separate experiments that demonstrated interaction between this protein here and that protein here. We can check what those things are by just trying to highlight them. If you click on a line, then you can highlight it. If you want to select more than one, you click with the shift pressed, and then we should be able to get two, and then three of those things selected. If we now look at the edge table, we do have three entries in the table. Each of those is corresponding to one of those three lines here, and there's the information that came from the interaction record. Among the multitude of columns that you see here, there's gonna be a column telling us what type of interaction was detected in a given experiment. There are two experiments that demonstrated that those two proteins are in direct contact, that protein chain of that this molecule is actually in contact with that molecule. There was another experiment where interaction was detected but was not certain if those two molecules are actually in direct contact, or maybe there's something that is a part of a bigger complex that mediates interaction between those. In such cases, the interaction type would be reported as physical. So this is one of the useful columns, and then there's information where the data came from. There's gonna be abbreviated list of authors of the paper. There's gonna be publication ID. The number here is PubMed ID. This is identifier of the paper the data came from, that can be used to find the paper in PubMed. And just to demonstrate it, I can just I don't want copy, I want to copy, and then I can try to see what do I paste in PubMed. I just want the number here, and this is the paper that actually describes the interaction that we've got represented as a line. So this is a way to find where the data for this particular interaction came from. Okay, now there's gonna be information about how the interaction was identified, what was the experimental method that was used to identify the interaction, and then there's gonna be a slew of columns with more data, more information about the particular experiment. We'll get over what's actually in those columns in a moment. Just to finish up as we did yesterday, once the interactions are imported from the Psychic server, as you've noticed, there's redundancy. There are those multiple lines connecting particular proteins. You might have interactions that start and end in the same place. Those are interactions, those are experiments that demonstrate that protein oligomerizes. So in case we don't need it, in case we want to simplify the network, we can simply go to, first we can unselect everything, and we can go to select to edit, and we can remove duplicate edges. This one is gonna remove edges that are connecting the same pair of proteins. So we're gonna remove those. We want to remove them from the IMAX network, from the one we're looking here. So I'm highlighting which network I want to work on. I'm gonna ignore edge direction. This is because in some of those interactions, edge might start at one protein and end at the other, but in some other record, the start and end of the edge might be reversed. So I want to ignore the direction. Just I want to end up with a single edge connecting two nodes. I'm gonna ignore the edge, but I want to create a table which is gonna list me how many edges contributed, how many experiments contributed to a given interaction. So I'm gonna check the box here, and now I'm gonna just okay. Okay, so we've lost 139 edges. It means that there were 139 experiments that were duplicated previous information. This is informative. The more experiments were performed in general, the reliability of information is higher. So the number of edges, number of experiments demonstrating given interaction tells us something about how good the data is. So that's the reason I created the extra column to actually give us a count of how many experiments contributed to a given edge. And now if we do okay, if now I go in the edge table and look what's at the very end, there's gonna be an extra column created here which lists the number of experiments that contributed to a given edge. We can sort on the column. The tables here, they work more or less like a spreadsheet. So some common operations are able. We can sort the column by just clicking on the column. Okay, and if we click here once more, where are we? We want to go this way. We do have we do have now one interaction with actually 10 experiments contributing to it, and then there's more and more interactions with less and less experiments, and then finally there's some strange thing which I don't know what it is, where there's no number provided. This is strange. It shouldn't happen, but I think it might be the number of interactions that were removed. So if there's nothing here, it means there's only one, versus 10 means there was additional 10 interactions, experiments provided. So that's, I think, might be the actual result of whatever the operation did. Now, oh no, this is actually not true. Let's see. This is, let's see. Oh yeah, this is actually not true. So if we check here, so those are the number of interactions that were contributing to a given edge. Okay, we are left with several entries without any number created. Now, if we look what those things are, we can either stare at the names to figure out what's going on, or we can actually select all the rows without any number here, and we can select all of them by holding on shift and clicking to select a group. And now once the rows are selected in a table, we can convert those into selected nodes, and then we can right click and go to select edges. Okay, and here we've got the suspicious interactions selected. And now if you look at those, you'll notice that we now have selected the edges that are looped. Those are edges that didn't get removed by the operation. So surely there's no number entered here, because nothing was done for those operations. In our case, I'm not going to be interested in homoligomers, in interactions between copies of the same protein. So I'm going to get rid of those. One way is to go to select, and then to edit, and to go and remove self loops. So this one is now gonna, once more, I'm gonna select IMAX as the network I'm working on, and then when I click on OK, the loops should go. And here we remove those. Okay, so that's our imported network from Psychic. So that's how one can generate networks by querying Psychic databases, and retrieving the records there, and then cleaning it up. Now, as you've seen, we're getting way more when querying Psychic than whatever we had in our simple file. This one, in our simple file that we initially used. The file that we were using had just two columns with two interacting proteins, interaction type, and that's about it. There was nothing there, no information for individual edges associated with them. So if we go back to the initial network, if we check the edge table, all we've got here is just the names of the interactions and the type. So Psychic provides us a lot more of a lot of more information. Now, what it is, we can check by looking at the by looking and going through the slides that I made for today. Let's see if that's it. Yeah, so that these are our slides. So we're gonna switch to slides for a moment. Okay, the PowerPoint is in on a Google Drive. It will go on UCLA box today in afternoon, so you have access to that. So let's see what's go into the records that Psychic is getting us. Well, in general, what's there? There's information about experiments that are used to identify that two or more proteins interact with each other, and the description more or less follows a very generic description of experiments that are performed to identify interactions, and normally what we've got to do first is we've got to make proteins in one way or the other. The proteins somehow got to be made. They might be made natively in a cells that are normally made. So we might be looking at yeast actin made in a yeast cells without any modification, just natively expressed protein. We might have this protein, the gene for this protein cloned and then expressed in E. coli. We might have the same case with human proteins that are made either in E. coli or in yeast cells or somewhere else. So there are different ways of making proteins. So part of the record will report where the proteins were made. There might be information how much of this protein was made. Was it made at the native level, just like it's made in unmodified cells? But in many cases, proteins are overexpressed or underexpressed in rare number of cases. So there might be information about that. Proteins might be made without any modifications, but they might be modified. It's pretty common to add a tag either in front of the protein or at the C terminal of the protein to simplify detection of the protein or simplify purification. So there might be modification made for technical reasons. There might be modifications that correspond to mutants that were observed in disease, and then one may ask question: does the mutation affect the interaction of this particular protein? So to check that, the protein would be made with those mutations present. Some proteins might interact only if they are modified in certain way post-translationally. Some proteins will interact only if they are phosphorylated at a given position. So there might be information provided in the record that, oh yeah, this interaction happens only if a given protein is phosphorylated at serine at position whatever. So there's potentially a lot of information that can be provided for each of the proteins. Okay, so this is the first step. We were making proteins. Then we've got to get those things somehow interact. In some cases, it's as simple as making them together in the same cells without any modifications. Just we can look at natively expressed protein in human cells or knee cells and look for interactions between proteins without any modifications to them. But it could be that one protein is made, let's say, in bacteria, in E. coli, purified, placed on a chromatography on affinity column, and then a cell lysate prepared from human cells is poured over that. So then we've got one protein made in E. coli. The other proteins are made in human cells, but then they are given a chance to interact only when we mix them together on the column. So then this is a different experimental setup compared to all those proteins made together at the same time in the human cells, and the records will reflect that there's gonna be an extra field telling 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 us where the interaction had the first had the chance to occur. Okay, once we brought cells together, the proteins together, we do have to test which of those interact. Okay, so this is actual interaction experiment. This is actually the method that is used to identify that interaction happened, and there's an entire slew of those. There's a lot of different ways of testing if interactions happens, and the record will interaction record will mention what method was used in one particular experiment. And then once we identify a group of proteins that interact, then we quite often identify once more what those proteins are. The reason is we might have a case that in where we make one protein first, we know what it is, but then we pluralize it from some cells over it to see what sticks to our known protein. And then once we identify those sticky proteins, we've got to identify what they are. So in some experiments, those proteins that were identified to interact do have to be their identities got to be confirmed after interaction is determined. So there's gonna be a set of entries in a record reporting how each protein was identified, was known actually to be present in a given interaction. At the same time, we might identify post-translational modifications that the protein was observed to when interacting. So there's gonna be additional information about the proteins that are interacting. This is very abstract in a sense, but very generic. Nearly every interaction experiment can be described this way. If we go into details, let's see how this thing goes into records. Well, each interaction essentially is composed of a button of a lot of pigeonholes. There are different places that are used for storing information about different parts of the experiment. Going through our flow of experiment, when we make proteins, we're gonna have a field for each protein that is gonna list where the protein was made. This can be different than the place the gene for the protein is normally coming from. There's gonna be information about the native version of the protein. There's gonna be information that we're working with a protein with such and such identifier that is normally made in human. There's gonna be information about the symbol for the gene or for the protein. There's going to be some description of the protein. There's going to be information that we're looking actually at interactions between protein, not between a gene coding for the protein. Some databases report just protein interactions or interactions between proteins. There are databases that report interactions between protein and the genes. So then and DNA, meaning DNA. So then some molecules might be actually reported as DNA, or in case of interaction with RNA, it might be RNA. So in general, the molecule can vary. So there's a field specifying what molecule type we're dealing with, and this is information about the native version of the protein. So this is information where the original gene of the protein came from, what it is. There's a cross-reference to a database that allows you to some database allows you to identify what the protein is. And then there's also information about the version of the protein as used in experiment. So one of the entries there is going to be where the protein was made. Okay. And then we will have, once we've got the, oh, and this thing goes for every protein that is reported to Interact. So if there are five different proteins, then there's gonna be five sets of information like that, each one corresponding to one of the proteins. So for each protein, you're gonna have information about native version of the protein and then where it was made. Once we've got all those proteins, we've got to put them together, and there's gonna be a field somewhere in the record reporting where the proteins had a chance to come together. In this particular case, in vitro means that the proteins were made separately in different cells and then brought together to observe the interaction. Okay, just as a short explanation, if we've got, let's say, three proteins, blue, yellow, and green, all made at the same time in the same cells, let's say in yeast, then experiment host would be yeast or cerevisiae. Those proteins might be originally coming from yeast, but they might be coming from human or from mouse or rat. Experiment host tells us where they came together. Same case, we can have three different proteins, all three made at the same time in the same E. coli cells. So then the host would be E. coli. If each protein is made separately in different cells, let's say there are three different E. coli strains, each one making individual protein, and then those proteins are purified and then mixed together, then the host would be in vitro, because the first time those three proteins met is actually a test tube. Same thing if we make two proteins in yeast, third protein is made in E. coli. Those E. coli proteins are this E. coli protein, or the protein made in E. coli, is purified, and then cell lysate from yeast cells is poured over column which bound to the yellow protein, then the host would be in vitro. So this is the meaning of experiment host that's reported in the interaction records. Now, this is, in some cases it goes against whatever experimental guys are using commonly when talking about interactions. It happens, those discrepancies happens every now and then when dealing with interaction records. So it's worth to be aware that some of the terms that you're gonna see and some of the fields that you see in interaction records might not agree with what people doing experiments are actually using. So in case of this experiment host, there might be some discrepancies, but as long as database records goes, the annotation is reported this way as just described. So this is one thing to be aware of. Now, once we made protein, once we got them together, then we've got to test if and how they interact. Okay, so there's gonna be a field specifying the method. In this particular case, it's a pull down. It's one of the affinity chromatography methods of identifying interactions. I'll show a couple of examples just in a moment. And then there's gonna be interaction type reported. Okay, in this particular case, interaction is direct, and typical things that you might see there are, those are the most common four terms that you're gonna see. Direct interaction means that if you take a list of interacting molecules. So let's say we've got yellow protein and green protein and blue protein. So if for every pair of yellow, green, green, blue, and blue, yellow, the interaction, the experiment demonstrates the molecules are in direct contact, the overall type of the interaction will be direct. If we've got homodimers, homo-oligomers of some sort formed by the same, by many copies of the same protein, interaction is also going to be direct because we've got only one type of a molecule but two copies, and there's at least one copy with direct contact. So it's also going to be direct. It's not always the case. It's not always that experiments can provide you with that level of detail. In many cases, all that experiment can tell you is that we've got a group of, let's say, three proteins that interact, that form the physical complex that can be isolated, and components can be identified. But experiment can't tell if we do have a case like here where every pair of distinct proteins is in direct contact. We might have a case where blue protein binds to green protein binds to yellow protein, and we can, and this is enough to isolate the complex like that. But it doesn't tell us if the blue protein is in direct contact with yellow one, because there's not enough information coming out of the experiment. In such cases, the interaction is going to be annotated as physical association. It means that the proteins listed as interacting do form a one particular physical complex, but we don't know what's the topology, what's connectivity. Another case where physical association might be reported is when we identified two proteins that are interacting, but we might have other proteins present in a complex which we didn't test for. This might happen, and then we know, we can only, experiment demonstrates that the red protein and yellow protein are present in the same complex, but they might be bridged by this yet unidentified protein that's providing a connection between those two. If experiment can't exclude those proteins bridging interactions, then the annotation also would list interaction as physical. So uh, that's how physical association term is used. And there's even a more generic case where we can tell we can experiments where experiment generates a list of proteins that are interacting, but all we know is that one particular protein is present all the time in possible complexes, but other four proteins like here may or may not be present at the same time. This is typical result of affinity methods, where in this particular case the red protein is binding specifically to some sort of a support, some sort of a resin in a chromatography column, or some sort of a solid support like in ELISA tests, where the red guy specifically binds to the support. Then a mixture of different proteins is poured over it, and then proteins that are retained are identified. We might identify the so-called bite protein. This is the one that binds specifically to the support, and several others, but we can't tell if all of them are present at the same time in all complexes. We might have a situation like here, where in some cases we do have red, yellow, and green protein retained on a column, red because it's binding directly to the support and to other proteins binding to it. But in some other places where the red protein binds to the support, the place of the green protein is taken over by the blue one. It might be that once the green binds, the binding site for the blue protein is blocked. So then they can't bind at the same time. Or it could be that they use the same binding site. They are very similar, they are homologous proteins that compete for the same binding site, and we might have a complex with either blue or green protein present, but never both of them showing up together. We might have also a situation where if the blue protein binds, there's another protein binding to it, so it will be retained, but only in a subset of complexes that do have the blue protein present. And a typical affinity experiment cannot distinguish between all those variants of the complexes that are present on a column. All those methods can identify is the list of proteins that was retained. One of them is gonna be playing a special role of so-called bait. So this red protein is always going to be present in a in those potential complexes. Other things may or may not. So in those case, interaction is going to be reported as association. It means that the list of the proteins corresponds to proteins that are present in one or more complexes that were identified. It only guarantees that every every so-called prey protein, although other proteins other than beta are called preys. So all those other proteins do have to be in a physical complex together with bait. So for this complex to be retained with the green protein present, it's got to be a part of the physical complex here. But same thing here, the blue and yellow and red thing, they form physical complex. Same thing here with four proteins. But we can't tell if green protein is present together with the yellow protein. Here, yes, it is. But here, no. Here we don't have, in this particular variant of the protein, we don't have green and yellow protein together, so we can't tell that there's any particular interaction between green and yellow protein. They might never never be present in the same complex. Here it happens they are, but it's not always the case. We've got a case here between blue protein and green protein. They are in a list of preys. Green and blue protein are present, but they are not never present at the same time. The affinity methods in general can't tell you what's the relationship between preys, and that's the reason to put them and try to annotate them as associations. So that's the least specific term which describes the interaction type. There are also more specific terms in some cases. Experiments can demonstrate that proteins are actually forming interacting through a covalent bond. The most common case is disulfide bridge between two cysteines that are bridging they are forming a covalent bond between two protein chains. It can be either for homodimers or for heterodimers. So in those cases, when experiment demonstrates the presence of a covalent bond between two chains, the interaction type might be covalent. What I did not list it here is entire slew of interaction types corresponding to enzymatic reactions. You can imagine that there's a experiment where we test if a given protein kinase phosphorylates some other protein, and if if experiment demonstrates that once we mix two proteins together, one of them becomes phosphorylated, then only in presence of the other one, then the interaction type for such interaction would be phosphorylation reaction, and for other types of reactions, there's an entire slew of terms describing those. So we might have more specific terms most commonly used for reactions. The most common terms those are the four listed here, with probably predominant physical association reported for the reactions. Now, uh, interactions like here with between three proteins or between five proteins, between three proteins here, do not go into cytoscape, because in cytoscape all the interactions are binary. If you noticed, the graphs are always the lines in a net in a cytoscape. The edges are connecting only two proteins. To convert to convert experimental data reporting physical interaction, physical association between three proteins or association between five proteins, we are performing. Oh, one notice: the association term is contentious. Some databases, some people don't use this term, and it's relatively common to stumble on people referring to associations coming from affinity purifications as physical associations. It created a lot of problems in the past. The good thing about IMEX databases is that they're using the annotation in a very consistent way, using the annotation as I described now. So once you're getting the IMEX records, you're gonna be you should be pretty sure that interaction types are corresponding to whatever is shown in this table. Okay, so now going back to interactions that are connecting multiple proteins, like here and here and here, uh, we've to in order to get them into cytoscape, they've got to be converted into pairwise interactions. This is so-called binary expansion. This is an operation which converts a record or converts a record listing five proteins as interacting uh, as association into a collection of pairwise interactions that can go into cytoscape, that can be presented as a graph. The for associations, the way it's done is according to so-called spoke model. What it means is interaction between one bait protein and several preys is converted into a series of physical associations between bait and then each individual prey. This is consistent with what we've seen here, that in every case of the complex hanging of some sort of a resin or some sort of solid support, every prey is guaranteed to be present in the same physical complex. So the pairwise interaction between prey, the yellow prey and red bait, is gonna be physical. Same thing with the blue guy and red guy here. Those two things got to be in physical. We can't claim interaction is direct because of the case like here, where we might have something bridging interaction, some other prey bridging interaction between the blue guy and the red guy. So we can't expand into direct, but we can expand into physical. So this is the case for the association. In case of interactions that are physical, we can convert them into pairwise interactions, each of them physical, So for the three proteins here, we can convert those into interaction between blue and green, blue and yellow, and green and yellow. This is once more because interaction is gonna get annotated as physical. Ah, where did I go? Sorry, where I'm at. Physical. Uh, yeah, this is because we are guaranteed that all those three things are present in a physical complex, but we can't tell from the experiment that we're dealing either with the case like here or the case like here. So we cannot claim that that interactions are direct. We can only claim that those binary interactions are physical. So so physical association is expanded, can be expanded into pairwise interactions between all the possible pairs. So this is so-called matrix representation, and the resulting type is going to be physical. In case of direct interactions, we are guaranteed that we're dealing with a case like this. So then every pair of proteins is in a of distinct proteins is in their direct contact. So then direct interactions can be expanded according to matrix model into series of pairwise interactions that are direct as their type. So so this is how this is how psychic is handling interactions that are originally reported as between multiple proteins. It's gonna generate for you the pairwise interaction so that they can be imported into Cytoscape as pairwise interactions. So that's how binary interactions are created from records that originally report interactions between multiple proteins. Okay, so that was a longish explanation what goes into interaction type, and then we're getting to the last stage to information how the proteins were identified, and then there's gonna be field for each of the participants listing one or more of the methods which were used to identify if what proteins were present. In some cases, the protein is known to be there before we do the experiment. The simplest case is we purified one protein, we purified the other protein, we mix them together, we observe that they form a complex, and we know it's got to be between those two proteins. So then we don't have to re-identify the proteins, and we specify that identification is predetermined. It means we knew what went in. But in other cases, one or more proteins are identified in separate ways. So then we're getting additional terms specifying how the protein was identified. And this seems to be a lot of information for each individual record. It might seem that this information is not relevant, but in reality, it does help to guess the quality of the data. It helps to establish even given experiment is more reliable or less reliable. On a scale of one experiment, it might be not that useful for one given experiment. But if we collect statistics over many experiments performed in many experimental setups, then it's possible to infer some conclusions about which methods are more reliable, which interactions are more reliable. So that's the reason a lot of the information is present in each interaction record. It might be not of interest to you because you're not going after evaluating different experimental methods, but still the information is there if someone is interested in looking at the different aspects of the experiments other than you might be interested in. So that's why some of this information is provided. Now, in terms of identifying proteins, the simplest case is we know what it is. In many other cases, we've got to identify those things in some experimental way. Here is a simple, relatively simple example. Those are three pull down experiments where one of the protein was used as a bait. This is the protein here present, then here, and then here's another bait present here, and yet another bait present here. Those were the proteins with some additional tag that was used to attach the protein to the chromatographic column, and then the proteins that bound to were retained together with a given bait were purified, and then were identified as sticking, and then they were identified by in this case mass spec. Okay, and the authors provided a list, let's say for this particular purification, a list of identifiers for individual proteins. Now, one might think that, well, there's an identifier here, so we know what the protein is. Let's if but if we think for a moment how the mass spec identification works, we'll see that it can be more complicated. So typical mass spec experiment, mass spec identification, at least one of the common approaches is, once we've got a gel like this with separate bands corresponding to individual proteins, this band is cut out from the gel. The protein is digested with trypsin into smaller fragments. Those fragments are separated on some sort of a chromatography column, typically reversed first column, reversed phase column, and then fed into mass spec. Mass spectrometer is gonna identify different fragments and will provide you the size of those fragments, and then in the next step, each of those fragments can be actually split into smaller parts, and this is enough information to actually identify what's the sequence, at least of a part of those peptides. So out of the mass spec experiment, you're gonna end up with a list of peptide sequences that were present after digesting this particular band with some sort of a protease, and then those peptides are used to search a database listing all the proteins in a given organism to find out what the protein is. The authors here came out with the identifier Q15906, and they claim this is a protein called YL1. If we go there, we're gonna find out that this is ambiguous. This is a fragment of UniProt entry corresponding to the identifier the authors provided. And if we go into the section of the record telling us about the protein sequences, we're gonna find out that there are actually two alternate versions of this protein. There is a one protein that's 364 residues long, corresponding to this Q15906-1 identifier, but there's also alternatively spliced protein with its separate identifier with -2 that is a little bit longer. And our mass spec experiment can't tell which of those two forms we're dealing with. This is why the identifier here doesn't have a dash. This is entry that's ambiguous. We know that maybe one, maybe the other, or maybe both forms are present, but we don't exactly know which one. So it means that in general, the identifiers provided for a protein can be ambiguous. They might correspond to a mixture of different variants of the same protein. And once more, depending on experiment, in some cases we might know exactly which isoform we're dealing with. This is typical for experiments where we're working with the cloned proteins made in some artificial system where we do know exactly which variant was cloned. So then we're gonna know what the variant is. We might have a cases when there's no alternatively spliced variant. So then we know that there's only one version of the protein, so we know what we're dealing with. But there are cases like here where there are two or more variants that might be present. So this is something to be aware of. Uh, it's also worth to be aware of that in case of UniProt, we might have two splice forms reported as isoforms, or two isoforms reported with their own identifier with the -1, -2, there might be -3, and so forth if there are more. There might be also additional potential splice forms present that were identified computationally. They will have a different different accession, different identifier, and those things might be present also in a sample provided they are made, but they are typically not reported. So there's always a chance there might be additional form present in the experiment if the experimental data is not specific enough. So this is one place where ambiguity between of the results pops up. This is similar in flavor to ambiguity between interaction types, where some experiments tell you that interactions are direct. Some experiments tell you the interactions are physical, but some can't tell you which of those are actually observed. So not all experiments are equal. Some experiments provide you more information, some provide you less. The records provided by IMEX are trying to keep track of that. So that's one issue with identifying proteins. In some other cases, like here, we might have protein identified as histone H2A, but if we check the record for this particular protein in UniProt, we're gonna discover that there are multiple entries in RefSync database, and it turns out if one checks those that this is actually a case of a protein that has 1, 2, 3, 4, 5 duplicated genes that each is making the same protein. The protein sequence is gonna be the same. The protein is gonna be the same no matter which gene makes it. But some but it might be made, at least when expressed natively from the cells that the genes are actually coming from, it might be expressed by only one of the 5 genes there. It depending on experiment, depending on what you're interested in, it might matter, it might not matter. In case if you're looking at interactions between proteins and you don't care about how the regulation of the expression goes, and if you don't care which gene contributed to the protein pool present in the cells, you might don't care. But if you're looking at experiments where you're looking at expression of the proteins at the level of mRNA and the level of individual genes, then it might matter that the proteins you're looking at, the expression is coming from different genes. So this is another issue to be aware of, depending on an experimental setup, depending on the information you're interested in. This might matter, or in many cases it doesn't. But it's worth to know that there might be a case like that. So this is another case of ambiguity. Here's ambiguity between protein and the gene that is actually contributing to expression of this protein. Now, in many cases, especially in case of structures, it is convenient. It helps to in experiment for the experiment to substitute protein from one species with a protein homologous protein from another species. So you might have Frankenstein interactions like the one here, where you've got different proteins coming from different species. If you track the actual protein identities of the individual chains reported in a structure described by this PDB record, then what you're gonna find out is that the beta 2 adrenergic receptor came from human. So this green protein here is actually human protein, but then the trimeric G protein that's binding to it is itself a Frankenstein. One chain is coming from cow, the, another chain is coming from rat, yet another chain is coming from cow. So depending on experimental setup, you might want to, once you retrieve the experimental record that describes this physical complex, you might want to substitute the bovine version of the yellow chain with the corresponding human version, and look at use this thing in further analysis. The same thing with the rat or cow. Or you might go and revert back to looking into bovine protein. So then the human protein will be substituted with the corresponding bovine version. You might have, for some of the interactions, reported additional molecules. There's a camelid antibody used to stabilize this complex, which is just a chemical used to stabilize the complex. It's not part of the interaction that we're looking at. So if molecules like that are listed, they might have to be removed from the further analysis. This is just experimental aid. So you might stumble on molecules like that listed as interacting. They should be removed. Sometimes additional domains or additional proteins might be fused with one of the interacting proteins to help in experiments. In this particular case, the T4-phage lysozyme was actually fused to beta 2A to simplify experimental handling of the sample. So this is another case which might show up. So there are cases like that. So this is another thing to be aware of. In most of the cases, most of those cases will be reported in the interaction record. So the information is there. It's just a matter of being aware what's there, and then making decisions. What should be retained, what changes should be done, what should be tossed, what information should be ignored. So this is something which depends. It changes on a case-to-case basis, but I'm just listing things that one should be aware of. And now finally there are special cases I'm gonna browse through, just breeze through. The interaction records do not report interactions of the proteins that are result of somatic recombination. Most common cases is antibodies, B cell receptors, and T cell receptors. Those are proteins that are made after the original gene as in a as in a genome is modified in individual cells of the immune system to make modified versions of antibodies. This is where diversity of antibodies is coming from, and those interactions are essentially not curated. So you shouldn't see the interactions for antibodies listed in the interaction records. And it also means that when you're interested in interactions of between antibodies and antigens and things like that, then you have to look into more specific resources, and you might end up on your own. There's not that many databases that handle interactions for particular antibodies. It's the reason is there's a no reference database that I know of that lists different variants of antibodies. So there's no way to curate those things in a generic way. I've already mentioned another case where we've got gene duplication and identical protein made by several genes. The most common cases you've seen was histones. If you stumble on those, you might have to look for a database dealing with specific cases. There might be database for particular type of proteins you're interested in. For histones, there's a database, at least used to be a database dealing with histones specifically, and then there might be some extra information there. Uh, and then another case is for viruses where in many cases viral proteins are made as a one very long chain as this guy, which is coming from one of the HIV strains. So the original gene lists is coding for a protein that's like 1400 residue long, but once it's made, it's chopped up into bits and pieces, each of them working as on its own as a separate protein. This is done by viruses to make the coding in a genome, a viral genome, more efficient. Viruses are limited by the size of the capsid, so they can't have too much genome package in a capsid. So they are trying to make it in an efficient way to provide coding information in a very efficient way. So one way is to make a long chain, long chain protein, and then chop it into bits and pieces. So here we've got matrix protein, capsid protein, another nucleocapsid protein, protease. So there are several different parts. In those cases, the identifiers will be provided like this. There's gonna be the UniProt identifier corresponding to the parent polyprotein, and then it's gonna be succeeded by a dash and this PRO and the number which identifies one of those particular things. So this is most commonly used for annotation of the viral protein. So it's worth to be aware of that, because other 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 data suppliers or might not use the nomenclature like here. They might use some different variant of referring to those individual proteins. So then those identifiers got to be somehow translated from one variant to the other. So this might be a problem in some cases. So it's worth to be aware and worth to know what identifier like this might look like. And once more, it's common for the viruses. And then the last pathological case is ubiquitin. Ubiquitin is very common, as the name implies. It's very short, 76 typically residue long protein that is used to tag proteins either by getting attached once or by attaching a chains of ubiquitins to a particular protein, or maybe putting it on at different places on a target protein. They are typically the attachment goes uh, through the C-terminal glycine binding to one of the lysines on a target protein. So this is how the connection looks like. Okay, and then ubiquitin itself does have lysines, so those chains can be built by attaching ubiquitin on a at a certain position another one, so things like that can be created, and depending on how the modification looks, it might mean different things. The most commonly used case is that proteins are tagged with ubiquitin for degradation. Once protein is ubiquitinated, it's directed towards proteasome and degraded. Protein is very common. Protein is very conserved. It's observed it's present in many organisms. It's not present in any database listing proteins that I know of, which is sort of strange. But the reason is it's not made as a separate protein to start with. When you look into a human genome, you're gonna find out that there's only a gene, there are only two genes that are coding concatenated fivemers of ubiquitin. They are made like the proteins I mentioned before, as a longer chain and chopped into bits and pieces, or they are made as a fusion with the ribosomal protein plus an extra ubiquitin. So there are two genes like that in human. There are three genes like that in yeast. And once those fusion proteins are made, the ubiquitin is chopped off and then starts to function on its own. But this short version, 76 residue long ubiquitin, does not have an entry. So this is something to be aware of. Because of those problems, there are two approaches. One version is to use a very obsolete entry in UniProt that refers to this individual 76 long residue ubiquitin that used to be present about 15 years ago in UniProt, and then it disappeared. So there's still this obsolete term used for annotation, or ubiquitin is annotated as PTM, as post-translational modifications, not reported as interaction at all. So depending on the project, you might want to either ignore all the interactions with ubiquitin, and this might be the most common case. But if you're interested in interactions involving ubiquitin specifically, then you might want to go after ubiquitin, even listed as PTM, and then convert those PTMs into interactions. So it depends on the project. This is one more thing to be aware of. So so that's the last pathological case, and then we got all the way down to the last part of the interaction record. So apart from identifying what the molecule is, we might have listed one or more protein features which tells us how different was the version of the protein observed in experiment from the standard version reported by in a UniProt or some other database. Those are typically mutations. Those are modifications of the sequence that were observed in a given experiment, either de novo, or those are mutations introduced to check what's their effect. There might be information if the protein observed in the interaction was phosphorylated, methylated, if it was ubiquitinated. And then there's gonna be a slew of features reporting what are the affinity tags used for useful for purification, what might be labels used that help to detect a given protein. In case of cross-linking experiments or proteolytic modifications, there might be report the position of the cross-link or position of the proteolytic cleavage might be reported. So there's a chance for additional features reported for a given protein. And once more, there might be none in some cases, but in some cases you might have multiple proteins listed in a record, and then for each of those you're gonna have one or more features. So that's why the interaction records are pretty complicated. And once the record is created, it's actually converted into presented in some sort of a file. As you've noticed from the description here, it doesn't fit a table format. This is not a description that can be listed, can be provided as a list of columns and with separate values. The reason is we've got many proteins or many molecules that interact. So we might have, we would end up with varying number of columns if we want to put information about one protein in one column. So for binary interactions, we would have two columns or two groups of columns. If we've got 10 proteins interacting, we'd have to have 10 groups. So this is not the tabular version. And even within a single protein, there are places where there are there are pieces of information that might be present in one, two, or more copies. So this is another case where this tabular format breaks. This is the reason what we've seen in the import from Psychic that's so messy. There are the Cytoscape, the Psychic is trying to pack all this information into a lot of different columns, sometimes providing a list of things within each column field. So that's why the table for the edge we've seen it looks complicated. But in case of records as provided directly by the database, it's much better organized. The standard format, here's a representation of the standard format, is called MIF, and it's sort of a nested Russian doll version where there are fields nesting down deeper and deeper, and there's hierarchy of how things are organized that mimics more or less the structure of the record here. So I'll here's the issues to be deal with, to be aware of, just to recap whatever I was mumbling for last half an hour or so. So here are things to be aware of. That one thing to remember is that depending on experiment, there's gonna be different bits and pieces of information present. So this is something to be aware of. And the second thing to be aware of is that in some cases the information coming from experiments is ambiguous, and that's why some of the fields in the records might have very 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 detailed meaning, but some of the information might be more blurry. It's not the problem with the records themselves. The records are able, databases are able to provide very detailed information, but in some of the cases, experiments are not providing that. So databases are providing only what came out of experiment. So those are the things to be aware of. We're gonna make a break now for 5 minutes, and then then we'll start playing with going after the records and how to read those things using Python. So now we've got the break for a moment, and let's get any questions if there are questions there. There's no questions. If I mean technically, there's way more about the experimental setups, about experiments that I mentioned here. For those of you who want to get more and didn't take the intro to Cytoscape workshop, I provide way more information there. You can go to the QCB site and get the slides from there. There's gonna be a pointer to two classes run at MBI and Chem Biochem department. One is typical. Both are run in winter quarters, so they are run at this moment. One is the class run by structure group. This is M230B and D, which provides a lot of information about structural methods that identify that can be used to identify a lot of interactions. So this is one class that's worth mentioning if you want to take it one time or sit through. It's very informative, quite quite entertaining. And then there's another class that's run by Emil Reisler and Jose Rodriguez and a few other people that goes over many different methods that identify interactions, and it's also possible to sit through the class without enrolling, just to listen to whatever is there, and then it gives you overview of the plethora of different methods used for looking at interactions. The Emil Reisler class does have a lot of invited speakers who specialize in different methods. So you're gonna get different segments corresponding to different methods. So it's a good overview to get an idea how experiments are done, and it's useful because it puts a lot of meaning into the records that you normally pull in from through Scikit or from other database, and then you can somehow put together the record and get an idea how actual experiments are done. The records are based on experiments, so it's useful to know how things are done. Okay, I'll shut up for 5 minutes, and let's get reconvene at 10:55, 5 to 11. So let's stop for a moment. And if there are questions, just ask, or either ask or type in a chat. So 5 minutes break. So let's continue on. Okay. So in the meantime, I put in one of the files that can be obtained from the IMAX databases that report that describe interaction records. The format used is in general XML. This is the native format that the records are originally prepared. By XML, what it means is it's essentially a format where you've got those uh, pairs of entries, pairs of tags, one with the name, the other one with the name preceded with a slash, that themselves might contain more entries that look like more elements that look like that. Some of them might have attributes attached to them in the name, and inside there's more and more stuff. It's like a Russian doll where you start with uh, one so-called root element at the top, and then once you unfold it, you see that entry set contains entry, and then entry contains source, experiment list, and so forth, and then each of those in turn contain more and more information. In case of our flavor of XML that's used for describing interactions, there's gonna be within entry set one or more entries. In this case, there's only one. Typically, entry corresponds to interactions coming from a single paper. There's gonna be always an element called source that provides information where the record came from. In this particular case, it came from the IP database, which is run here. So there's information about the source with a lot of details there. Once more nested down. So there's a group. There's an element called names providing short and long version of the name of the database. There are some cross references providing pointers to papers describing the database and so forth. So there's source. Then there might be a list of experiments provided here, once more containing one, in this particular case, description of experiment. Then there's a list of interactors. Those are the list of reference proteins that might be used in experiments. So once we unfold this thing, we do have now 1, 2, 3, 4, 5, 6, 7. A lot of interactors here. Keep going. That's a lot. Okay, we've got a lot of interactors here. If we unfold just one of them, we're gonna have a lot of stuff here, but in general, you're gonna have information about what's the name of the protein, either as a short label, there's a description put into full name entry, and then alternative names of the protein. You might have one or more, typically a lot of cross references to various databases. There's always identifier in a given database, and then name of the database. So this is cross-reference pointing to UniProt KB database entry with identifier ID Q12165, with additional information here, plus a lot of cross-references to other pieces of information. Then there's gonna be information, what's the type of the interactor. So in our case, it's gonna be a protein. There's gonna be both name of the type and also cross-reference to a controlled vocabulary entry, which defines what the protein is. So there's gonna be cross-references here. There's gonna be element that describes the species that this protein came from originally. This is where the gene from for the protein is coming from. So in this particular case, it's the yeast protein. This is the taxon identifier, species identifier, according to NCBI, plus the short and full name of the species, possibly with an alias. And then there's a sequence of the native protein. So this is what you typically find as a description of a reference version of the protein. So that's what's showing up in tractor list. And then we've got also list of interactions, and now we've got two of those. If you look at one of them, there's gonna be one or more cross-references. In this particular case, there are cross-references pointing to experimental data deposit in protein data bank. So there are those cross-references provided to us pointing to WWPDB here and here, and identifiers are PDB codes. Those are the identifiers of the structures deposited for this particular interaction. There might be additional cross-references there. There's gonna be a pointer to experiment, to information that was provided higher up. And then there's gonna be a list. Let's see. Then there's gonna be, let's fold this one. Let's fold this one. And then there's gonna be a participant list. Let's fold this one. Let's fold this one. So for a given interaction, we've got the name of the interaction, and we've got cross references to experimental data typically, and there's gonna be a pointer to experiment. So this is gonna be a pointer to experiment description showing up here. And if you quickly check here, there's the information about where about the paper the experiment was described in. There might be cross references to databases also describing this interaction. There's gonna be host or organism or experimental host. This is the experimental host you've seen in description of the record. So this block describes where the proteins had the first chance to meet together when interaction was observed. There's gonna be description of the method that that was used to identify interaction. In this particular case, it's electron tomography or 3D electron microscopy. This is cryo-EM essentially, and there's a pointer to the method. Okay, then there's gonna be possibly a description of a method used to identify participants that were observed to interact. So there might be an entry like this. There might be some additional information provided as an attribute list, and all this block does have an identifier here that's then used as used in experiment ref to refer to the block above. So that's the second piece of information in the interaction. And then finally, there's gonna be there's gonna be interaction type. If we unfold it, we're gonna see that this is interaction that's physical association. and it's provided as a set of short and full short label and name and full name, and also a cross reference, also as a cross reference here to a controlled vocabulary term, to a formal definition of what the method is. So there's gonna be a cross reference like this, and then so this is interaction type. There's some extra flags specifying if this is actually interaction that was modeled based on some other species or not. So this is the case of the complex I've shown you with the mixed species. If the record describes interaction after conversion into human, this flag would be set as true. Uh, so there are a set of flags specifying details like that. And finally, there's gonna be a list of molecules that participate in a given interaction. So if we start folding those things, there's gonna be many of those because it's a big complex. And if I just look at the first of those, for a given participant, there's gonna be pointer to one of the interactors showing up earlier on the list. So this is the reference to the standard reference database version of a protein, and then there's more information about how the molecule, how the protein was used in a given experiment. There might be information about so-called biological role in case the enzyme was used, the protein was used in experiment as an enzyme. Might be instead of unspecified role, there it might be specified as an enzyme, or if the protein was a target of an enzyme, it will be enzyme target and so forth. So there might be different things here. Under biological role, there might be a description of the role the protein played in the experiment. So this is where you might find the information if the protein was a bait or a prey. Once more, in a standard way, there's gonna be always a pair of names and cross-reference to a controlled vocabulary term. This is where the term is defined. In most of the cases in the record, you're gonna see a limited set of fixed values that are standardized, so there's no ambiguity in terms of using using different names for the same method. It's all this information is standardized by using sets of terms that are well defined with their own identifiers, so it's easier for programs to deal with those things without confusing names used in by different people. So most of the terms are standardized here. So here it's a list of one or more roles that protein is playing in a given experiment. There might be information about this, how the protein was prepared. There might be information it was purified or not purified, if it was overexpressed or expressed at the physiological level. So there might be a block of entries like that. And then finally, there's gonna be a list of features, if there are any. So here in this case, there's information that this particular protein was used only not as entire thing, but there was only a part of it used in a given experiment, and there's pretty complex way of reporting what is the region that was used, and this is all sitting under feature and there might be one or more features. They might correspond to regions of proteins used in experiment, mutations, and so forth. So this is pretty complex thing sitting there. We already, oh, there's gonna be a host organism. This is where the protein was made. This is for the participant. This is where the protein was made. This is different than whatever you've seen, whatever you've seen in experiment. In experiment, there's host organism. This is the entry here. The value here corresponds to where the proteins used in a given experiments were first put in contact. So this is experimental host. When you look at the host organism for the participant, this is where this particular protein was made. So those are two different things. The value here and value in the experiment might be different, and they've got different meaning according to whatever I described for the record. So here's participant, and this is the first one, second, and so there's many of those. Then there's, I mentioned, I think there's gonna be, for a given interaction, there's gonna be information about the interaction type. So in this particular case, it's physical association. So all those proteins are present within the same protein complex identified by cryo-EM, and because of that, interaction type is physical association. So it also means that we either don't know, or if every two proteins are in direct contact, or it might be that we know that only some of them are in direct contact but not all. So interaction associated annotated like this can be always split into pairwise binary interactions that are physical. So those two participants can be annotated as interacting and forming physical association, binary physical association, and can be done for every pair taken from the list here. Somewhere in the participant, there's gonna be hidden information about stoichiometry. At least it should be. If it's not, well, it's attribute. I don't see it, but there should be information about stoichiometry if present. So this would be also accessible here. So information about how many copies of a given protein contribute are present for a given interaction. So information also might be somewhere in a participant here. So here's interaction type, the flags I've mentioned, and then this is one interaction with, and here's another one. The difference is, well, let's see, what is the difference? I think there's no difference. Might be a different version of a complex with its structure. So we've got another interaction here. So anyway, that's the structure of the interaction record presented as a file. As you can see, it's relatively regular structure. In a sense, it's a nested set of nested elements, starting from the one at the top, and then we're getting deeper and deeper into the file, and we're finding more and more detailed information about different things. And we'll spend the rest of today trying to learn how to read files like that. It's useful to know, to be able to process those files, because the format is very universal. The details of what the names of the elements might be different, the meaning of different fields might be different, but the structure is common to many records. We can, for example, check UniProt, and we can look for, let's see, P6001, or we can look for gal 80 yeast. Okay, and if we look for the entry for gal-AD protein in yeast, we got it here. This is the standard page you're gonna find on the on a Uniprot site. But if we add to the URL .xml, now we've got the representation of the Uniprot record presented as an XML file. Once more, the structure of the entry is very similar to whatever we've seen before. We've got the top level element and then something nested here, in this case, the actual entry in a Uniprot, and then copyright info provided here. And then inside entry, once more, we've got a set of nested things that provide the contents of the record. What you see as the name of the elements is different than whatever you've seen from the interaction record. The information is different, but the structure of this file is very similar, and it turns out it's very easy to read with a script, and it is very easy to access different bits and pieces of information. So let's start with trying to generate one of the files we used yesterday to annotate our graph. As a reminder, what we had yesterday in Cytoscape, we took one of the files that I've provided, and we generated this column with the gene name corresponding to the formal name of the gene in our network. The network originally was created from a file that listed only two two genes and nothing else. So we had to get the human readable gene name from additional file, which was given was provided from outside. So let's try to get a file like that from scratch. Okay, so to do this thing, we're gonna fire up Spider. If you installed Anaconda, then you should have somewhere on the desktop, or somewhere, if you're on a Mac, somewhere in a Finder, you should be able to find Anaconda Navigator. If you start it up, it should open your window like this, and then you can go to Spider and launch the Spider. Once you start it, you should end up with the window that looks like this. Initially with just the test. With just the test file. Okay, and to start with, I would suggest that you make sure that if you go to Tools and Preferences, you set a couple things here so that your Spider behaves the same way as mine. One thing to go is to go. So once more, to get here, I went to Tools, and get me chat so that I see if you're okay. Back. So once more, if we go to Tools and Preferences, you want to go to Python default. You want to go to Run, and this is okay. interface appearance 7. No, this is gonna be an editor. Yeah, so you want to go to editor in preferences. You want to go to run code, and you want to make sure that all three things here are checked. So then you should see roughly the same thing as things that show up on my in my spider when I run anything. Once you check those things, just do OK, and it should disappear. Okay, and then once you've got spider up, then you can go and load the file that I dropped into. I've dropped into Google Drive. This is the one. This is this one to download. The one ending with pi. Okay, so let's look what's there. Okay, so to deal with XML files or with XML with data presented as XML, we've got to use a special module, special library that provides a lot of functionality when dealing with XML data. This is the line that's gonna load the module. The library is called LXML, and I'm importing only one entity there called E3, and I'm naming it as capital ET. This is pretty common practice. Then you can refer to any method, anything that library provides by referring to et dot something. Okay, so that's what I'm doing here. Okay, and then if it okay, and then in case of reading files from the network by providing URL, in most of the cases the URLs are provided with secure encrypted HTML URLs, so using HTTPS. For XML to be able to deal with it, we need additional library, additional module imported. So this is something that's needed so that we can get to files that are coming through encrypted HTTPS. So those are two things that typically show up there. And then in Spider, we can split our file into so-called cells. So they are split by, the code is split into cells by using /$. So if I type $, I'm now seeing a single cell highlighted, the one I'm in. And then if I want to execute code inside, I can press Shift Enter, and the code here was executed, and what I see here in the console is output what the code generated. In my case, nothing much happened except for seeing the copy of the code popping up here, and this confirms that the imports work. It confirms that I do have the libraries present. If you do see errors here at this stage, scream, tell me, then it means you might have not those two libraries imported present in your Spider installation. So then I'll help you with that. But in general, this thing should work, and this module shouldn't complain. You shouldn't see any errors here. So that's those are preliminaries. Now what we need is a sample piece of XML. So here's a cell that defines a very simple valid XML file. I'm just defining a variable my XML with triple single quotes. This thing allows me to specify a string that spans multiple lines with whatever I want to put in there, and then I terminate it with another set of three single apostrophes, single quotes, and whatever is in between is my string. As you can see, it's composed of a top level element called record. The element goes all the way to the very end. So entire string is composed of one element starting with the record opening tag, and then it's closed by slash record. Those two versions of the tag are present all the time. There's always the opening and closing tag, and then inside there's more of those. There's an element called protein nested inside, and there's another one nested inside. As you see, it starts with the tag called with the protein name, ends with the tag with the slash preceding protein. So this is one element. This one has an attribute called ACC with a string as a value, and it does have another element nested in named SEQ, obviously with a fragment of a sequence corresponding to whatever protein we've got. So this is very simple, simple version of a XML file, XML record that we can deal with. So when I run this piece of code once more, I just press Shift and Enter. I'm gonna see now the print statement here, printing out what's in my XML, and here's my printed version of what's in my XML. Now, XML is very generic, very generic. It can be used for different applications. So there's a chance that the names of the tags used for one applications are exactly the same as the names used in some other applications. For example, someone might have a tag called name, and there might be a name sitting there, but in one application the name might be a name of a person, because let's say this is a snippet of XML that provides that keeps track of data in someone's phone book. Another name might be name of a protein, because this is a piece of XML that describes actually protein entries in UniProt. To make it easier to keep track of different applications of XML format to different types of data, there's a notion of so-called namespace, and here is a little more complicated version of an XML file. Okay, this is a snippet that starts like an actual record in a interaction record. Okay, we've got the top level element called entry set, and here's the closing element. The thing to notice is that now the name of the element does have a prefix mif dot colon something. This version of an element name means that the entry set is coming from so-called a namespace, and namespace itself is defined by an attribute at the top level. Here, it always looks like XML and S colon, and then there's a prefix here that is then used for all the other elements. And the attribute here means that every time a name is with the prefix mif, it's coming from a namespace, HTTP colon / and so forth. Very often this URL might be a place where the particular flavor of XML is defined, but not always the case. And we don't care what's sitting under the URL here, under the namespace here. It's sufficient to know that that prefix here means we came from a namespace defined at the top level here. There are also three attributes here, and then there's a bunch of things nested here. And as before, this snippet goes into a variable called mythxml ns. And now when I run this block with shift enter, now I see the string printed out. So this is our second case for the myth files. Now, how to read it? So far, we just define the snippets of XML and printed them out without knowing what's inside. How to get our script actually start getting after individual elements? Well, it's deceivingly easy. All we've got to do is we've got to call a method called fromString and pass to it the name of the variable with the holding our XML. So, and then the result is stored in my XML DOM variable. Let's see what happens. Oh, and et means that this fromString function method came from the et module that we just imported at the very top. So so here's the one-liner that actually reads my XML variable that were with the XML string, and then when I run it, then I'm printing the type. I'm just checking what's in the variable here. So I'm just printing the type of XML DOM. If I do type my XML copy and paste type. So here I'm printing type of my initial string and then type of whatever the method returned. Now the first argument passed to print was a string. The second one is this horrible thing that apparently came was defined in our library. This is the library that we imported, and this is something that was defined there. So to go back from whatever fromString returned and get back the text representation of the XML record, we can just call another method provided by the library called toString and pass whatever came after parsing. So here I'm gonna have to type actually my XML DOM. So this is what was returned when we were parsing the string. I'm passing it to the function, the method that reverts the parsing operation. I do add this decode thing because whatever toString returns is actually not the string. So I've got to add this thing. This is peculiarity of Python and how the strings are represented. So I've added this decode here. It would work without, but let's keep it here. And then when I run it now my print et returned me whatever was in a string. So so far we didn't go into detail. So far we didn't do anything other than just printing the contents of the file back. The useful thing is that we do have our data in XML present in a variable here. In a moment we'll see how to get to different bits and pieces there. The convenient thing about the library is that we don't have to provide a string as a to to the to the library to read it. We can specify that we want to read a file. So here's the path that I've got to a file called myxml file.xml. This is this file here. It contains something like this here, and the file is sitting on my desktop. Uh, it's sitting on my desktop down down in down in PPI here and here. So this is the file. I can just under Windows, I can just find the file, do copy, and then paste the name into the file. And this is where how to refer to the file in the script. Okay, this is so-called fully qualified path to the file here. There are ways to make it shorter, but without going into much detail. This is the simplest way to get the proper pointer to the file on Windows. So here's my file. So now instead of calling from string, I'm gonna call a parse method. I'm gonna pass a name, the location of the file, and then once I call parse, then I'm getting the another variable with the contents of this file. So I'm printing it here, and let's see what happens. I run it, and here's the contents of the file returned after first parsing it into a data structure that our library generated, and then converting this data structure back to string. So that's what we've got here. Even more entertainingly, LXML allows you to actually access files that are not local to your PC. So this is a file sitting on my hard drive somewhere. We can pass actual URL. We can pass a location of the file on the internet. So here's one of such locations. Okay, let's say copy, and let's paste it here. And when I go here, I do have a file. I do have an XML, piece of XML, but this is piece of XML that my browser pulled from the UniProt. This is equivalent. This is a record that describes the UniProt entry with the accession, not source, with the here. So this is gal80 entry in UniProt. When I add to the URL here .xml, I'm looking at the XML data downloaded from UniProt, but now I can just use this URL, provide it to parse. Because there's HTTPS prefix, I've got to do it using URL open. So I've got my URL sitting here as a string. I'm passing this my URL to URL open method, and whatever this thing returns goes to etparse. Okay, and now once I run this block, Control Shift and Enter. Now you notice there was a slight delay. This is because my PC went to UniProt, pulled in the file over the network, and then printed it out. So the response might be slower because of the network speed. But essentially now I've got exactly the same thing that you see in a browser when accessing one of the Uniprot records. So this is sort of convenient. We can parse either local file, we can either local file like here, or we can read and parse record provided somewhere on the internet. So this is kind of nice. We don't have to do anything else, anything special. It's nearly identical operation. As an alternative, we can always read this file into a string and pass it to a from string. That's another version, but in all cases it's relatively simple, and in all of those cases we're gonna end up with something which either parse or from string method returns, and this is gonna be the contents of the record there. So now, how to get to individual bits and pieces that are in the file? Well, so far we didn't go. So far we were just reading the file and then spitting it out. So it's not that useful. But let's try to go into bits and pieces. Okay, so here is my XML DOM. So this is who knows what, but I'm just printing what's sitting in my XML DOM so that I know what I'm dealing with. Then there's three dashes to separate, and then first thing I can do is I can print a tag property of this guy, and this will return me the top. Let's this let's run it first and then see what happens. Okay, so here, so I just pressed Control Enter and run this snippet of code to see what's going on. And here I'm printing the MyXML DOM in a text representation. So this is what's printed out. Now I've got those three dashes to mark the end of the file. And then next print statement is printing out MyXML DOM tag. So this is gonna be the tag used for the top level element. So we do see record printed out here. This is the name of the top level tag. Then I'm looping over the element over this MyXML DOM. This is gonna get me one child element at a time. So during the first pass through the outside loop, I'm gonna get to this protein element. Then during the second pass, I'll get to the second element. If there's more, I would get more of those. So here I can scan, I can go element by element over whatever was present in the record. Okay, so that's what's going on here. I'm printing them. Then the tag for whatever is the current element I'm looking at. So this is here. I'm getting protein when I'm passing for first time through the list of children of record, and then second time I'm getting once more protein. This is for the second guy. I do print then the list of attributes that this element might have. Okay, to get to the attributes, I can just specify keys. In this context, each element behaves as so-called dictionary, and dictionaries do have a set of pairs. Dictionaries are collection of pairs of a key and a value. in our case, keys are gonna be names of the elements. So the ACC is gonna be a key in a dictionary, and P600 is gonna be value, and this is gonna be dictionary associated with the first protein element, and then there's another protein element. It will also have associated dictionary with ACC as a key, and then the value for that is gonna be the P04 and so forth. So here I'm just printing the keys. So this attributes here and child.keys allows me to find what are the attributes for a given element. And here I've got attributes returned as a list with just one ACC as its element. This is because I've got only one attribute here, and then I'm going after value of this attribute by calling a get method, and I'm passing the name of the attribute here, and then I'm getting here the attribute value. So I'm getting, when during the first pass, I'm getting P60010 here, and then during second pass, I'm getting the value here. So this way we went through the protein element and then another protein element to get deeper in, to go after the things sitting inside protein. We can repeat the procedure. We can look for nested children of our current element. So child as here corresponds to either the first protein or the second protein. So now I can ask what are the nested children of my child. So once more, this is another loop now going over whatever is in child. So I call those things N1 child, our nested first level or whatever. And then when I'm going through this loop, once more, I'm getting the tag of the child. Okay, so now this is gonna be SEQ. This is gonna be the SEQ here, and then I want to get text sitting inside the element. This is another variant of what can be in XML file. I get just a piece of text bracketed by the opening element and closing element. To get to this thing, I can ask for a text associated with an element, and this is what's printed out here. So so this way we can get to any element in a XML file. So as you can see, it's extremely easy. This is all you need to do to get through XML file, to get through the data provided to you as a XML file. So that's the reason to use XML. It's much easier, at least in my opinion, than trying to go after different formats, because you're getting things. You can get to things in extremely simple way, and it holds on for any type of XML, as long as the overall shape arrangement of the file is compliant with XML. You can get to anything that's there by just looping over elements and looking for either attributes or more elements or texts sitting inside. Still, it is somewhat complicated, because in order to get something nested down here, you've got to manually descend through entire hierarchies. You've got to find what's the top level element, then you've got to loop over the set of children, then for each of those you've got to loop once more, and if there are more levels of nesting, then you end up with not two levels of loops, like two level of looping here, but many more. So there's an easier way. There's an easier way where what we can do is we can specify so-called XPath, which tells us what we're after, and the call to XPath method is gonna get us a list of things that we're interested in. Okay, so the way it works is, first you can notice that to specify that we want to find a text like this, here we've got to say that we start at the top of the file, we want to get a record element, then we want to go one level down and find the protein element, and then go one level down and find the seq element, and then get the text sitting there. To specify that, we can just write a string like this. It starts with the slash, meaning we're starting at the top of our document. We're looking for an element called record. Then we're looking for a child of the record with the name protein. Then we're looking for a child of a protein called SEQ, and then we want to get the associated, and then we want to get the piece of text sitting inside SEQ. So this thing corresponds to descending through the nested set of elements down to what's here. Okay, and once we specify something like this, we can just call this method called XPath. It's gonna interpret the string and then try to find what's matching. And if we run it, now what I get as a result is I'm getting now a list with two strings. The reason is there are two locations in a file that match the search. We can either find this piece of sequence sitting in the first protein element, or we can find the second sequence sitting in the next protein element. So in general, XPath can return more than one value, and because of that, it returns a list of things. So you see, this is not a single string, this is a list. You can tell it because when it's printed, it starts with the square brackets, ends with square brackets, and there are two elements there corresponding each to one of the sequences. So thing to remember is XPath is always going to return a list, even if it finds only one thing, it's always going to return it as a list with just one element. So what goes into XPath can be pretty complex, and this is the simplest case where we're going after, like, we want to descend 3 elements down and find the text there. If we want to put some restrictions on what we're going after, we can specify so-called conditions at some level. So there's an example here. What I did is I put in square brackets a condition on a protein element. What I want to do is I want to get a protein element, but only with attribute called ACC. To specify attribute, I say at here ACC. This is the way to refer to an attribute, and I want to make sure that this ACC is equal to 560010. So now this XPath is gonna match. It's gonna find elements matching in our XML data structure only if we start with record, we go to protein, but only with ACC attribute set to 60P60010. And once we've got match here, then we're going down to SEQ, and then we're getting text. So now This version of an XPath is gonna get us just a single record corresponding to accession P600110. This is kind of neat. You see, you don't have to iterate, you don't have to write loops. You just specify a path like this, just one string, and you can make a beeline to whatever piece of information you want to get, and you don't have to bother with extra stuff in a file, extra things that you're not interested in. You can read a file with your record, and then you can get to individual piece of information just by specifying the XPath. There's there are ways to make this those strings even simpler. There's a way to use wildcards and arbitrary nesting. Here, when the path is specified this way, we want to start from the top. Then there's got to be a record element at the top, followed by protein with whatever, followed by seq, followed by text. If But we can say that we want to look for any protein element starting somewhere in a file. We can present somewhere in a file, we can just specify dash dash. This means that we've got arbitrary elements above protein. So in our case, we've got record, but if protein was sitting two levels down, then this expression wouldn't find it. This one finds it just fine. We can also specify that we want a piece of text sitting inside an element with arbitrary name down below protein. So this thing is also gonna work. As we run it here, we're getting once more the same string. The difference is here we specified explicitly the entire hierarchy. Here we took a shortcut. We just decided we're going after every protein element in a file with a given accession, and then we want to get the text in every element nested one level down inside protein. Those are examples, a little bit more complicated XPath expressions. There's many more features. They are coming. They can be specified. There's a description of those in Wikipedia. There's a link here to check what's there. Okay, copy, and if I paste it here, there's a web page describing what's there, and there are examples of different things you can do with XPath. So this is for your reference, and there's also a rather dry specification of exactly what's the syntax of the XPath strings is. It's pretty dry, but this is the ultimate reference to find what can be put there. So that's how to deal with files that have no prefixes, no namespaces. As you see here, this is a piece of XML with no prefix, with no namespace here. We can deal with files that do use prefixes. It's very similar to what we've done here. The only difference is now is that we do have to specify what namespace we're interested in. The way to do it is to add additional argument called namespaces to our XPath function call, and we put an arbitrary prefix that we want to use that's associated with the namespace we expect in the file. And from now on, we can put a prefix in front of every element that belongs to the namespace here. So let's run this thing here, and now we've got the file with the namespace called mif here, and here's the full name of a namespace. I'm lazy here, so I'm using just the prefix M instead of MIF, which corresponds to the same namespace. Okay, so this is the same namespace here as here. And then once I specify that M corresponds to this, I can now use it as a prefix to find an element called source and then find an attribute called release date. Okay, and then once I run it, I do get the release date printed out here. It's only one value, but XPath returns always a list. So there's a list with one element here, and I got to the element here. So this way, we got the release date for our first interaction record. That we parsed, or at least part of a file that looks like an interaction record. So that's how to deal with the with the namespace XML file. And I'm gonna stop here because we're just over time. So I'm gonna stop here, and we'll wrap up tomorrow by trying to generate this file for the adding a label to the stickers in Cytoscape, and then we'll try to read a few XML records using a library that builds on top of XML. So that's what's gonna happen tomorrow, and I'll drop this file. This file is actually present in a in a in a in a in a Google Drive, so you should be fine. I'll also put it on your CLA box, so for your reference. So that's about it for today. So hopefully by tomorrow you'll be able to read not only the the simple files here, but also the records, and add them to the Cytoscape. So we'll see how far we'll get in tomorrow. Okay, any questions? And if you can't get Spider run because you see any errors when running this piece of code, then let me know. I'll figure out what's your problem. If you do have any questions, I mean, email me or complain down or ask on chat, and I'll help you to get this thing going. So it's no problem with that. So that's it for today, I think. Any questions? No questions. People are disappearing. Okay. Okay, so thank you everybody. The recording is gonna end now. Okay, so let's stop recording. Yes.